Anyway, it's a huge pleasure to be here. Edwin, thank you very much for the invitation to participate. Anna, your presentation this morning reminded me of how complex the issues are that we're dealing with, and I'm afraid my presentation is really focused on just one dimension of it, and it's focused on the dimension of trying to provide humanity with a deeper understanding of the diversity and distribution of species on our planet, to build a more intimate relationship between humans and other life forms. Of course, it was very intimate in the past, and it's been eroded as we've moved into urban settings. So our work is designed to bring people back in contact with nature. We're living in an extraordinary time in Earth history. There have only been 5 prior mass extinctions, and we're living at a time when the next one could occur. And you are the community who are going to contribute the knowledge. I mean, humanity is not going to be interested in living a lonely life with strand organisms alone. I think we're going to want to have real DNA-based life forms with us. They're really interesting. So, how can we do a better job of understanding the diversity of life? And that's really the focus of my presentation. The International Barcode of Life has been advocating for the use of DNA-based approaches to accelerate this process. BIOSCAN was a project that it launched earlier this year, and its goal is to lay the groundwork for an Earth observing system for species. Now, DNA is not going to be a complete solution, obviously not. We're going to have diverse approaches, and clearly satellite-based imaging is really powerful. It's going to provide us a synoptic view of things like algal blooms, forest loss, shifts in the phonology on a very large scale. So this is useful. But when it comes down to essential biological variables, biodiversity variables, we're not going to manage biodiversity from space. So these are the 12 EBVs for the ocean. And the color coding indicate the green, they indicate those which are going to require ground-based or sea-based surveys, and the blue indicates those that can be remotely sensed. And you'll see that all of the variables will benefit from Earth observation at ground level, and there are 5 that will benefit from remote sensing. So we're definitely going to need to build a very large and sophisticated Earth observation system. How are we going to set about doing this? Our planet's a tiny one in the universe, in the solar system, but it's big enough to be reasonably complex when you ask, I'd like to know everything that's living everywhere on the planet's surface. So as a first beginning, one might consider partitioning our planet into 150,000 square kilometer blocks. And if you did that, you'd end up with about 1,000 divisions on land. Germany, that will give you 2.3 blocks. That's a pretty coarse resolution, I would say. So as a place to begin, what about considering a biosurveillance grid where those 150,000 square kilometer blocks are all broken up into grid squares of 1,500 square kilometers? And this is what Germany would look like under that model. These would each be blocks where potentially we could go and carry out biosurveillance. Here's an organism, one species of the many millions that share our planet. It's the emerald ash borer. And the green shows its native distribution, and the blowout to the bottom part of the curve shows what it would look like if we had this grid scale resolution, which of course we did not have in 1995. And we certainly didn't have it in 2005 when that species expanded its distribution into Eastern Europe and into North America. And we still don't have it in 2015 as its range expanded. The 2005 number was between 1995 and 2005 was really important. If we had had grid square resolution in North America, we might well have recognized the arrival of the emerald ash borer in 1998 or thereabouts, and have averted a $280 billion expenditure on the North American public. Think of a future. Think of a future where every species on our planet is registered with this level of resolution, grid square resolution of every species. And here's 3 of those species. The green is the native distribution, the orange is the invasive distributions that spread through time. Doing this recurrently, on an annual basis perhaps. Think about this as a weather system for biodiversity. It happened in the 19th century. We set up weather monitoring networks. We haven't yet done that for biodiversity monitoring, and it needs to be done. And not just distributions, but all the biological covariates associated with it. The size of the organism, the species with which they interact, their phenology. That's going to cost a lot of money. And a lot of money is not something that biodiversity science has traditionally had. We haven't been a member of the Mega Science Project Club. In fact, there's only one biological project that invaded that terrain, and that's the Human Genome Project. But you can see that astronomy, physics, space have been there recurrently, $150 billion to set up the International Space Station, which has a finite lifespan, and it's done great things, but that's a really big investment. Doesn't biodiversity science deserve to be on that grid somewhere? How did they justify these level of expenditures? Astronomy was there first, so it's an interesting place to look back and ask how George Hale convinced the Rockefeller Foundation in 1928 to invest in the construction of the Palomar telescope. No method of advancing science is so productive as the development of new and more powerful instruments and methods of research. Those 2 things, instruments, new methods. All of the subsequent mega science projects have adopted this philosophy. Large instrumentation, centralized facility, gathering data in a very organized fashion. It changes our perception of the universe. More energetic particles than we've ever seen. More distant, better resolved stars than we have ever seen. And you don't do this by yourself. You do it in a big team. 10,000 researchers involved in the Large Hadron Collider. 2,000 in the Hubble telescope. This is what we need for biodiversity science. But let's think about our discipline and where it's been. In terms of technological innovation, our discipline has relied very heavily on a single technology, microscopy, for centuries to tell apart organisms. It works, but it requires a very laborious process of human investment. The situation is changing. DNA-based technologies developed for medical applications are now available for use in biodiversity science, and they're doubling in their power every 9 months, twice the pace of Moore's Law. These instruments can gather phenomenal amounts of data. Hook that technology with the simple idea that you only need to read about 1/1,000,000th of the genome, just a few hundred base pairs, and you can tell apart all of the species that you encounter. Couple those 2 things together and you can do amazing things. And this is a point that we've been mentioned already today, and that's the decline. The World Wildlife Foundation, 60% decline since 1970. The wonderful study that came out of an amateur natural history society in Germany demonstrating the 75% decline in insect populations. The study 2 weeks ago in Science that documented a 30% decline in all of the bird species collectively, 3 billion missing birds in North America. These are really important observations. They motivate the public. I think if we have more granular, more volumes of this data, it's going to be very influential in causing humanity to take steps that we're not left with a lonely planet. But I do want to point out that where did these insights come from? A non-governmental organization, an amateur natural history society, and a small collection of researchers based in government and lab. These are artisanal. This is the results of artisanal studies. I admire them hugely. But this is not the future of our science. We need industrial scale analysis of life on this planet. The International Barcode of Life is trying to bring this approach, trying to scale up our capacity to analyze the diversity of life. Its mission is to implement an Earth observation system for biodiversity. Got a 25-year plan, and over that interval, it will register every species of multicellular organism on the planet. It will document their interactions, and it will monitor their populations. Those are all achievable goals. He'll achieve this through 3 projects. The first one was Barcode 500K, completed in 2015. $120 million expenditure. BIOSCAN was just launched in June. It's running through for 7 years, about $180 million investment, and then the large 20-year project, Planetary Biodiversity Mission, that's at least 500 million euros. Let me change to your currency. And that's what would require less than €1 billion investment to advance towards these goals. Barcode 500K, what did it do? It delivered an information service. It asked you to imagine a world where anyone could identify any organism that they encountered. And it committed to delivering sequences for 500,000 species in 5 years. And it did 4 things. It did deliver barcodes. It created a core facility that was running 1 million specimens per year. It developed the informatics platforms to store those data, and it advanced a system that allowed the automated recognition of animal species. iBOL then took a holiday. Well, not really a holiday. It spent 3 years looking for new plans and protocols that would make it possible to scale up analysis. So that's 2016 to 19. Let me talk about what resulted from that. 2 analytical paths, barcoding, and a barcode is you take a single specimen, recover a single short sequence from it. Metabarcoding, take a large assemblage of specimens, extract DNA, gather large numbers of sequences that are connected to that sample. The applications. Metabarcoding, 1 application, species occurrence data. Value, really cheap, a euro penny per specimen. Barcoding, you can build the reference library that you need to interpret the metabarcode results. You can identify specimens, you can discover species, species occurrence, species abundance, problem, €1 a specimen. These 2 workflows are benefiting from the tremendous advances in DNA sequencing technology. The Illumina platforms, the NovaSeq and the MySeq, are ideal for metabarcoding analysis. They generate very short reads, about 500 base pairs, but they generate large numbers of them, up to 500 million reads in a run. The SQL platforms generate many fewer sequences, maximum 3 million per run, but the sequences are 50,000 base pairs long. And you might say, we're barcoding, why do we need to know that? Because the error rates in these sequencers are quite high. So here's the workflow, the barcode workflow on the SQL. Take a specimen, extract DNA, gives you a DNA extract, you PCR, you chase the amplicon you're interested in, and you sequence. That thing called a smart bell, that's the circular form of the amplicon, and basically the sequencer runs around inside that smart bell multiple times. And so you get a very accurate sequence. One problem with this workflow, that little chip you see there costs €2,000. So it'll cost you €2,000 to barcode 1 specimen. But you don't do it like that. You mix up 9, you take specimens, 24 3D4 well plates at a time, extracts, amplification, but tag each one of those specimens up there tag 1, tag 2, and so on. Pool 9,216 specimens. Barcode them, and you get sequences recovered that are exactly the same as Sanger sequencing, but for a cost of about 1/100th what Sanger would cost you. That was Sequel 1. The new Sequel 2 platform has 8 times the throughput. It will run 73,000 specimens in a run. That single machine will analyze 40 million individual barcodes per year at a cost for the entire workflow of €1 a specimen. Here's the metabarcoding workflow. You start with a collection of specimens, extract PCR sequence, match the sequence to the reference library. You need to do that to get the specimen identification. You need that reference library. You don't usually do just a single sample. You do multiple, and you tag the amplification products from each DNA extract separately so you can go back to that sample, to that bulk sample. One complexity. You might think, okay, here's an example. Let me just show you the complexity you encounter. Here's a standard metabarcoding study, a simple one, 375 species. Kept simple, every individual represented by one individual. Pull a leg off, every individual, pull an abdomen off, bulk them up, extract the DNA. So it's basically metabarcoding, but we know how many species went in. How many reads does it take to find those 375 species? Answer, to get 95% of them, you have to do 100,000 reads. When you add abundance variation, which is something that happens in nature, of course, you need at least 1 million reads to recover those 375 species. Okay, that sounds pretty formidable. Yeah, it used to be formidable, but not with the new platforms. Here's the NovaSeq. It cranks out 500 million reads per run. That means you can run 500 samples in every one of these runs, and the cost per sample, all costs in, is 50 pounds. The sequencing cost would be about 10 euros of that total. And you think about what that single machine could do in a year, it can run 62,500 bulk samples a year, 1,200 sites, weekly sample from each of them, resolved. Species composition. This is the new big problem, and it's one that you guys are close to, and it's the volume of data that's being created. A Sequel 2 will generate 1.6 billion reads per year if used flat out. A NovaSeq, 62 billion. The species occurrence records that will be generated by each of these machines per year is 40 million off the Sequel 2, and the NovaSeq, if you assume 300 species on average in a sample, you're going to get 20 million time-stamped, location-stamped, species-identified records. Now the cost? 43 million to run a Sequel 2 flat out, 4.5 million to run a NovaSeq flat out. I doubt anyone's got that operating budget right now in biodiversity science. Large Hadron Collider operating budget is 1 billion euros a year. It costs 13 billion euros to discover the Higgs boson. I think biodiversity science can afford to do these kinds of studies. You need to do something with the data. And of course, we've been working hard, the iBOL community has been working hard to develop 2 platforms. One is the Barcode of Life data system, which has been around since 2004 and has been gaining power since then. And mBrave is the new platform that's been developed to deal with the flood of data from the high throughput sequencing platforms. It interfaces with BOLD. I want to talk about this, a particular aspect of BOLD, the bin database, because this is really important. As was mentioned several times, most of the species on our planet are unknown, dark taxa. How do we have a fast way to recognize all these unrecognized species? And the bin system gets us a long way towards what we're aiming for. So just as a reminder, if you haven't looked at barcode data before, This is a sort of topology that you typically see. Very little variation within a species, deep variation between species. That's an extremely useful pattern. So these are Australian fishes, marine fishes. These are insects in Costa Rica, Saturnid moths. And you can see the same kind of topology, this lack of variation within a species and deep divergence between species. So when you have a pattern like that, you realize that you can use methods to delineate the number of sequence clusters. And so that's what the barcode index number system is doing. It takes sequence data and inspects it and decides upon, in an automated way, the number of sequence clusters. So now we have sequence clusters. In a way, we've just automated species discovery if this happens to be some group of organisms which we have no taxonomy for, soil mites, almost no taxonomy for. So you immediately get account of the number of clusters, and the bin system goes one further. It names them. It adds a unique alphanumeric. And this is one of those bins. That's one of those bins, one out of 651,000 that currently exist on BOLD. And as you can see, it brings together images, it brings together distribution, it brings together genetic information, sequence divergence to the nearest neighbor, and the patterning of variation. This is within the species, and that's the nearest neighbor. And of course, connected to taxonomy as far as we can connect it. We don't want to walk away from Linnaeus. We want to try and drive into the Linnaean system as far as possible because there's a lot of useful covariates that have been collected over the last 250 years. But this is the fast way to register biodiversity anywhere on the planet. So you can go to sea, you can climb a mountain, you can go a cave, wherever you would like, and collect specimens. Make sure you got permits, and barcode the specimens, and don't throw them away. The genomic community has often been keen to get the sequence information, and what was that thing we got it out of? All these specimens have to be properly created and end up in a natural history collection, and then run them through the bin algorithm, and you have a species count. And where careful comparisons have been done in groups with really well-developed taxonomy, about 95% concordance between bins and taxonomic species. It's not perfect. What else can you do with the bin system? You can just press a button and you say, hey, I'm interested in knowing what fraction of the species are in common between southern Canada, Costa Rica, and Argentina. And you get the answer like that. Embrave, this other platform, I don't have time to talk about it. My colleague Sajeevan Ratnasingham is giving a couple of presentations on it. Please, if you're interested in what you do with these high volumes of data, make sure you're at his talks. It's a phenomenal platform for the use of this next generation sequencing. So that's the sort of preamble. Now let's get into BIOSCAN. What's going to happen over the next 6 years? What's this project going to do to lay this foundation for the planetary biodiversity mission? Well, first of all, it's going to engage in species discovery. It's going to analyze 10 million specimens. It's going to dig into species interactions. It's going to look at 5 million specimens for this. And then finally, it's going to do 100,000 bulk samples to look at species dynamics. The recent IBOL publication suggests there might be 8 million species, 1 million of them at risk. Very useful to have those numbers on the table. Attract a lot of attention. As someone working in the domain of playing around with DNA and species, we think the number is more likely to be close to 20 million, but that's a quibble, right? But 18.1 million of those are dark taxa. BIOSCAN is going to try and illuminate as many of those dark taxa as possible. We're going to need to do an awful lot of work on groups like fungi, an awful lot of groups on the hyperdiverse taxa like nematodes and mites and harpacticoid copepods. There's just a lot of those groups, huge numbers of undescribed species. The other thing we're going to do, though, is place-based analysis and using shallow skims, as we're calling them, where we go into a site, use a standard collecting method, and analyze 20,000 specimens. In this case, this shows you Malaise traps deployed at 183 places around the planet, year-long deployment, take 20,000 specimens, barcode them, not metabarcode, barcode them. And when we did that, the first 2.7 million specimens gave records for about 214,000 species. Now imagine that, taking that up an order of magnitude, and all of a sudden we're at over 2 million species. Aside from the skims, we're going to do deep dives. We're going to do deep dives in 3 places: in the Arctic, in Europe, and in Costa Rica. And deep dive, we want to barcode every species that occurs in these settings. It may only be 10,000 in the Canadian Arctic or in the Arctic. It's more than 100,000 species in Europe, that's for sure. And in Costa Rica, it's almost certainly more than 1 million species. And this is marine, freshwater, terrestrial, plants, fungi, animals. This is the tough place, Costa Rica, 1 million. BioAlpha, the deep dive. The idea is over a decade to register the 1 million species in that nation at 25x coverage. So we're going in 25 individuals on average, cost 25 million euros. This project has the support of the government of Costa Rica. That's the environment minister, and standing beside him is their president. And you can see this was the decree that said this is in the national interest to understand all biodiversity in our nation and be able to tell it apart. And this is the kind of stuff we've been doing so far, just on a tiny scale, looking at rainforest, cloud forest, dry forest, moist forest sites just a few kilometers from one another. And there's the species accumulation curve, 250,000 specimens analyzed, about 250,000 species, rainforest, more diverse than the other habitats. The thing that surprised me as someone who plays in the temperate zone mainly is just the lack of overlap between these ecoregions. There are exactly 32 out of the 17,000 bins that we collected in dry forest, rainforest, and cloud forest, and even less when we took the 4 ecoregions. So immense diversity in Costa Rica. We're hoping to wrestle it to the ground over the next few years. We're up at about 50,000 species based upon prior work we've done. And the plan is to do 1 million specimens in 2020, 1 million the next year, 1.5 million each year after that, and drive towards closure. And the green, happily 3 organizations, Costa Rica included, the Walder Foundation, and also the Guanacaste Dry Forest Conservation Fund, have provided funds to cover the cost to do the sequencing on those specimens. So at least the first bit of this, if any of you have a few million in your back pocket, please, fund one of the other years. Species interactions. Up till now, we've just been able to look at organisms to try and understand their interaction, but that's about to change. In many cases, we found funny things when we looked at organisms, things that fed on too many different parasites, like this one that attacked way too many hosts, this insect that fed on way too many plants. And when they were Sanger sequenced, we found that these were in fact cryptic species assemblages. So Sanger sequencing did deliver some interesting results, but there were many other cases where the Sanger results were uninterpretable because we got 2 sequences from them. If you analyze these with SQL, it's really cool because you now get the 2 clouds of sequences separated. You can interpret them. And most of the sequences connect to the mosquito or the biting midge or the chironomid, but there are these others that show really deep sequence divergence. And what are they? And when you take a look at those sequences, they're showing us the symbiome, the interactions of those species. This little midge was feeding on it was from Africa, happily. It wasn't from Canadian Arctic, and it was feeding on elephant. And this mosquito from New South Wales was feeding on a brush possum, swamp wallaby, a nematode inside the chironomid. We're at a point where we're going to be able to reveal interactions between species in a whole new way. And the methodology we're using so far just doesn't have the depth of sequencing, so we now have a new workflow that's cranking up sequence coverage. So you take a caterpillar and you hit it with all these different primers, and you can pull out with that primer the food plant that it had, these primers are going to pull out parasitic nematodes or parasitic wasps. You'll understand the constellation of species that were associated with that one specimen. And of course, that's only the beginning, because it becomes very cool when you don't just take one caterpillar, but you take several, and then you start to build up the interaction network around that species. And of course, that's only the start, because then you start to connect all of the species in the ecosystem. This is absolutely going to be, I think, a major growth area and a major exciting stuff. In our own species, these things are going to be complex. 329 species that parasitize us. 10% of them are only known from humans. So get ready for complexity. We've started work on insects. That's going to be an early thrust because they're tiny and you can work with them quickly. And we found, nearly 500 species of nematodes just in a quick scan that mainly were unknown. So let me move to the final topic of what BioScan is going to do. So it's species discovery, it's species interactions, and it's towards global biosurveillance. And I'm just going to be able to talk about two of the projects that we've undertaken. One is up here in Kruger National Park, the largest national park in Africa, and the other is down here in the smallest ecoregion in Canada, and asking, can we go into these places and survey life quickly? And here's Kruger. And the rangers oversaw this malaise trapping program for a year. It was fantastic, and they did a great job of it. There are two ecoregions in the park, and there was essentially no information on the insect community in that park. And in the course of 1 year, these samples were made. The samples finished collection in June of this year, and the results are all available today. We have records for 20,000 species. 80% of them were new to the BOLD system. There was a difference in species diversity between the low-veld and the Mopani woodlands, and the insect assemblages matched well with recognized ecoregions. So imagine doing this in every national park in the world and looking at how ecoregions are changing through time. This was green field barcoding, where it was a site we had no information. By contrast, in Ontario, we've been doing quite a lot of barcoding, so this is the area we're working in. We went into 45 provincial parks, did the same sort of thing, year-round, malaise trap sampling at those 45 sites, and counted the specimens. Well, we counted 250,000 of them, and estimated the rest. There were about 1 million specimens in those samples, and we barcoded 250,000 and meta-barcoded the rest. And the barcoded specimens delivered 14,000 species, but only 5% were new. We were sort of wasting our time doing barcoding. We shouldn't have done that. That was quite expensive, but we needed to make a point. And these are the meta-barcode results, and the combined results delivered data for 25,000 species, and it took 2 weeks of machine time. And because they're running 2 different machines, it took 1 week of machine time. And this is the data that we got from it. I can't easily display the linkages between all 45 habitats, but here's some of them. And you can see these nodes here. These are the bins, the species that were only found in a particular habitat. So if you have to prioritize habitats that you're interested in protecting or want to make special effort to, it's probably those with more endemic taxa species only found there. That's BioScan. BioScan is the beginning. It's laying the foundation for this big project, the Planetary Biodiversity Mission, this 20-year project, and we're ready to do it. The instruments are there. I wouldn't have said that a decade ago. The instruments are there. The informatics platforms are there. They need to be expanded and keep gaining new functionality. And there is a community of researchers that are prepared to put serious energy into this. So I'm confident by 2045 that we can have completed the registration of all multicellular life and established this global biosurveillance system. At the moment, there's a single core analytical facility following the model of other sciences, a single facility that specimens are being sent to, processed in an organized fashion, cost-effective, but that's not the future. The future is right here in Leiden, a European center, or let's have another one in neighboring Germany. Let's have one in Asia, probably a couple. Every continent is certainly going to need one, and I think just like we have cyclotrons today in virtually every country, we can anticipate that there will be large analytical facilities making use of this technology to read nature. Can we afford it? Absolutely. Humanity spends billions of euros every year to advance our understanding of the world. There's just no question it's affordable. And in fact, it's not just affordable, it's one of the greatest science bargains on the planet. 500 million euros to do the planetary biodiversity mission. The flyby of Pluto costs 600 million euros. The flyby of Pluto. What is more important to humanity? Okay. We are living at a time when life on our planet is imperiled. I think 15 years ago it would be fair, or 10 years ago it would be fair to say that we were ill-equipped to deal with the challenge. We had all these species that we did not know. Okay, that's still true, but it's not going to be true much longer. We could look at species interactions, but it was really time-consuming. And our capacity to map and understand the distribution of life was weak at best. In 20 years, things are going to be very different. All species will be known. All multicellular species will be known. We will understand a lot about their biology associated with it. We will have studied their interactions. This will be a powerful tool for advances in ecology and evolutionary biology. And finally, we'll have established a global biosurveillance system that's tracking life on our planet just like we track the weather today. It will be taken for granted. The work that I've just described is only possible because of a fantastic community of researchers around the world that have doing all they can to support this effort, the species skims. And the area de Conservación Guanacaste, Dan Jansen and his team there and the government of Costa Rica have been so fantastically supportive. It's just a privilege to be able to thank them. And then finally, in Kruger National Park, the support we've received from SANBI and other authorities in South Africa, and the researchers and the park rangers, has just allowed something that was just, hey, we were there, we had a Barcode meeting, we knew nothing about the insect life, and now all of a sudden we've got a detailed understanding. And of course, all of this would be impossible without the support of our funders, and so I'm deeply grateful to them. And if you had been in Toronto the evening that we launched the very first project, Barcode 500K, you would have seen our largest building in Canada, the CN Tower, with the DNA barcode of Castor canadensis run up it that evening. So that was sort of a cool evening.